Welcome, everybody. Thanks, Gábor, for the invitation, and thank you for the organizers. So, I will speak about the A540 lung adenocarcinoma cell, which was mentioned in the previous lecture as a very well investigated cell line. And that it is a very interesting cell line. So that, but before that, I will speak about the role of deuterium in water. So I speak about this from the quantum and thermodynamics implications. What is the difference between HDO and H2O? So the first thing is what I mentioned on the, I'm sorry, I have no pointer, I guess, yeah. So the first thing is the 0-point energy. That's the basis of everything. So you have a change in the 0-point energy that stabilizes the deuterium compound. The next thing is the enthalpy. So you change the enthalpy as the 0-point energy difference makes the energy different and the enthalpy also different. So you will have a difference in the in the energy, and that's, for example, you can see on the boiling point, freezing point. Other thing which is very, very important is the entropy. It's a huge change in the entropy because of the symmetry change. And all these together with the temperature influence the Gibbs free energy very importantly. So the idea is that the transcriptome is responding to this altered thermodynamic landscape. So the deuterium changes the thermodynamics of the body, and the isotopic perturbation that begins with vibrational physics and ends in the cellular phenotype. That was the basic idea. So briefly, I show here two figures together. On the left side, you see the barrier distance, so the hydrogen is tunneling easily. It has a lower reaction barrier that leads to kinetic energy effect, as the deuterium has a much higher barrier and much less tunneling. And on the right side, you see the potential energy curve, the two 0-point energy vibration, and you see the unharmonicity, which is very important, as you see the O deuterium bond is shorter compared to the OH bond. So here I saw how the symmetry breaking is influencing the autoionization of the water. So if you distinguish, autoionization is completely symmetric normally, but with water. But here, deuterium introduces three new compounds, HDO naturally, H2DO plus, and OD minus. I don't go into the details now because I think that's not that important. So here I briefly show you the vibrational landscape, how vibrational landscape changes if you replace hydrogen with deuterium. You lose symmetry, you obtain a new vibrational frequency. So I give you the numerical values there, but those are not important. The figure is important. And this calculation, this is a quantum chemical calculation. This calculation is in water. So, and now I focused on the cation, what we obtained, and you see the cation vibration is quite different compared to the non-deuterated cation. So our thinking started from this two excellent paper of Zubarev and colleagues, which was mentioned, so I don't speak about that more. So you know now what it's about, so I can spare some words. And another paper which was very interesting for this study was this messenger RNA expression of A549 under the deuterium stress. So they used a simple permissive threshold to overcome the measurement noise, because the measurement was noisy. It was a nanostring measurement, rather noisy, but they measured in duplicates. And finally, they eliminated the noisy data and obtained a quite simple result that at 300 ppm enrichment, nearly all cancer genes are overexpressed, 97% of them, and under depletion, the genes are stable, or some of them are underexpressed. So this was the basic idea. So what is our motivation? So I showed the biological logic behind this study. The central dogma defines a direction flow from DNA to messenger RNA to protein, meaning that any isotopic perturbation must first manifest in the messenger RNA expression. So the change of the deuterium concentration must represent itself in the gene expression. And this model, this A459 cell, has a very, very specific protection against any medicine or anything, so it is very stable, very aggressive. It's called KRES, STK11, and KEAP1 mutation. So we did two analyses. The first analysis was a different filtering of the noisy data, and we obtained at the end 87 genes. And in the second study, we used more refined, more extensive filtering, and we focused that we keep all the important genes in the study. So obtained 102 genes, and these together show that the biological signal is robust across methods, while the choice of error model controls the breadth of the transcriptional response, not the direction. Okay, so this figure shows you what is happening. So in the initial filter, when you excluded the expression with low counts, it's 150 ppm, we retained 87 genes, and rigorous filtering, we kept 102 genes. And these are, we hope, high fidelity genes. So we did our study on these genes. So this slide summarizes the conceptual framework behind our findings. Deuterium acts as a metabolic dial that shifts the cell between two opposing physiological states. On the left, deuterium depletion pushes the system into braking mode. Oncogenic signaling weakens, drug resistance circuits collapse. And the cells become more sensitive to therapeutic pressure. On the right, deuterium enrichment drives acceleration, crash-link pathways, intensify metabolic reprogramming, deepens and resistance rates consolidate. The central idea is that intracellular deuterium content is not a passive isotope ratio, but a regulatory variable capable of tilting the entire signaling and metabolic architecture of crash mutant lung adenocarcinoma. So this is where I show what is happening in this cell. You have the triple protection, 3 locks. I symbolize it with 3 locks. Here is the malignant core of the cell, and you see the crash keeps the proliferative engine permanently engaged. Through constitutive crash MAPK signaling, the second lock, STK11, this drives the invasion, the metabolic rewiring, and an immune-cold microenvironment, giving the tumor plasticity and escape capacity. And the third lock disables oxidative stress, breaking creative chronic NRF-driven antioxidant shield, and that neutralized therapy induces ROS. Overlaying these mutations is a wild-type TP53. Sorry about these abbreviations, but that's how this can be expressed. It's very dense, but maybe the pictures resolve this denseness of abbreviations. So you see this triple protection of the cancer cell, and this is why A549 is so good model. So if you can have positive effects on this model, then you are on a good place. And just a representation. So I show the signaling architecture of this cancer cell. So I show the core, and I show the pathways and the different genes, how these genes are collected or collecting themselves on a single pathway, and how these pathways are interconnected. So this is a quite complex machinery. And this is how we try to influence this. So the input is nanostring expression under different deuterium content. So we changed the deuterium content of the cell and measured the expression. 40, 80, as Roman mentioned, 80 is important, and 300. 300 is almost 350, but yeah, it's close. So we focused only on the transcriptome, and we used Roman's proteomic results. So in the first filtering, this is what we got. The situation is very simple. At 40 ppm, no strong upward expression, too moderate expression increase, stable 66 and 17, and too moderate or strong underexpression. At 80 ppm, the situation is somewhat similar, and at 300 ppm, you see the overwhelming overexpression. So now we continue with the 102 genes, the high-fidelity genes, and we use dbScan to identify the sentinel outliers, and we do clustering, how these genes are clustering under deuterium concentration change, how they reorganize themselves, because they have an attack by the deuterium, and the cancer cell will reorganize its expression. This is the basic idea. So this is the overview. On the right side, I show the pathways. On top, I show the deuterium content, and the colors represent the overexpression or the underexpression of the given pathway average. So I collected all the genes according to pathways and calculated the average expression, and that's, I color-coded this expression. So you see now more clearly that deuterium depletion leads to underexpression, deuterium enhancement leads to serious overexpression. This is a visual idea that gives you visual idea, several very important genes I show here. The average overexpression is 44%, but several key drivers like IL-6, TGFBR2, and DAPK1 showing the strongest individual amplification. But this is important because this is a coordinated escalation of activities of inflammatory, invasive, and survival pathways, simultaneously demonstrating that deuterium enrichment functions as a metabolic accelerant. Okay, and here I show several decreasing expressions. These are also very important. For example, ABC1 is the transcriptional amplifier, MYCN and the growth factor receptor is FGFR4. So all decline substantially. What this shows, this pattern demonstrates that depletion does not trigger indiscriminate damage. Instead, it selectively weakens the circuits required for resistance amplification and growth factor responsiveness, functioning as targeted metabolic brake on the malignant network. So the next thing is dbScan. You see the outliers here, but let me show you a more detailed representation in the three-dimensional space. You see here 40 ppm, 80 ppm, and 300 ppm, and in this space I identify the extreme responder genes. So these are inflammation, invasion, drug resistance, growth factor signaling, and transcriptional amplification genes. Okay, let me focus on one, this ABC1, which shows an interesting V-shape expression, and you see the target at 80 ppm. It's the expression is 42% decrease. Why is this important? Because this EBC1 gene protects the cancer cell against therapy. So if you try to introduce any therapeutic molecule, it cannot enter because ABC1 will immediately remove it. So it is therapy resistant because of this. But if you can, it cannot resist to deuterium depletion because the cancer needs water. It doesn't take it as foreign molecules. So this shows that the cancer cell will be sensitive, but only at 80 ppm. At 40 ppm, not. So if you continue to decrease the deuterium content, this effect will disappear. Now this is the final slide. So this is the 6 cluster we identified by the GMM6 method. And on the left side, right side, you see that, for you left side, you see that how 4 cluster is not enough, and you have to introduce 6 cluster to properly represent the answer of the cancerous cell to deuterium stress. Let me show you these 6 clusters, and yeah, crash-linked and TP59, so you can read it. So it is clustered according to functions, not randomly clustered. So the genes react in clusters to the deuterium effect. This slide highlights the cluster 1 and 2. The cluster 1 is cell cycle repair, which is very much overexpressed, so the cell will repair himself under this deuterium increase. Cluster 2 is the rush signaling engine, also protecting the cancer cell very much. This is the 3rd cluster when TP53 tries to balance against cancer. On the right side, you see the cancer, which is ignited, but TP53 tries to balance this effect. And here I show cluster 4. This is growth and stress response. Cluster 5, ECM remodeling and invasion. So if you increase this at 300 ppm, it's a very bad thing. If you decrease it, it's a good thing. And cluster 6 is the basal maintenance. Here I show you another effect. At 40 ppm DDV, you destroy one of the locks very clearly. So the cancer cell becomes vulnerable. So this is the main idea. This slide closes the loop between physics and biology. The mass-dependent vibrational stiffness alters the kinetic accessibility of the hydrogen bond network, reshaping redox flux across the isotopic gradient. At depletion levels, this constraint drives oxidative stress and suppressive transcriptional pressure. At enrichment, it relaxes Roche load and fuels activation and transcript. The transcriptome is therefore not responding to a chemical, it is responding to thermodynamic boundary conditions. Deuterium concentration sets the energetic landscape that defines oncogenic resilience. So this summarizes my talk, this figure. So it draws your attention that at 80 ppm something happens with the cancer cell. Something important happens, and it will become vulnerable. While at 300 ppm, it is very virulent and very aggressive. Okay, thanks. Thank you for the attention.